Hello everyone, this is Zainab Sheikh Muhammad, a teaching and research assistant at the Wayne University of Science and Technology at Egypt. On behalf of my colleague and my supervisors, I'd like to present our research project titled Predictive Analysis of SNP-disease Association for Alzheimer disease. Alzheimer disease is a slow progression of memory failure and language disturbance, poor judgment, and ultimately death. According to the World Health Organization's report published in September 2021, 55 million people have dementia worldwide, and it's predicted to reach 78 million in 2030 and 139 million in 2050. The pharmacologic current AD treatment do not necessarily slow or stop the damage and the destruction of neurons that cause Alzheimer's symptoms and make disease fatal, and that's why the focus of future medicine shifts from treating existing diseases, typically late in their progression, to preventing diseases before it sets in. The elevated risk of AD along with the potential AD preventable strategies that are recently invented create a crucial need to discover and validate prognostic biomarkers that may identify subjects at greatest risk of developing Alzheimer's disease. Although genome-wide association studies have been successfully employed to interrogate the genetic architecture of common and complex diseases, they are limited for being based on single locus analysis, and they fail to detect the epistasis interaction between different loci and their effect on the appearance of the phenotype of interest. The main aim of our project is to evaluate the impact of different selection techniques for selecting significant single nucleotide polymorphism associated with AD. We assess two approaches. The first one is the p-value SNP selection technique, which depends on the statistical significance of SNPs, while the second one is the interaction selection technique, which is a hybrid approach that relies on the ability of machine learning-based selection techniques to capture the cumulative effect of small effect size SNPs, which is variable in complex diseases. Then, by building a machine learning model for each chosen subset of SNPs and evaluating their accuracies, the power of each approach is to be assessed. And by revealing the genetic basis of AD, we will manage to build more predictive models for Alzheimer's disease. This is an overview of our proposed framework. First, our dataset was obtained from National Alzheimer's Coordinating Center. It consists of 364 unique samples. Each sample contains almost 380,000 SNPs. The framework applies a pre-processing stage according to specific quality control steps to ensure the data meet certain criteria, such as missing rate tests and imputation, the minor allele frequency test, SNP encoding to represent SNPs as numeric values. We divided the SNPs into 4 batches according to their p-values, and to evaluate the p-value SNP selection technique, the only SNPs that should be used as features are the ones in batch 0, as its SNPs p-value are 10 to the power of -5, while the other batches were used in testing the second selection technique. In assessing the interaction technique, we consider a higher number of SNPs to capture their cumulative effect. However, we have to keep the number of SNPs comparable with the sample size to assure that the training model will not overfit the data. Hence, working with a finite number of data samples in a high-dimensional feature space, we needed to eliminate some features using a feature selection method before using another model to fit the data. Here, we try to propose machine learning-based feature selection techniques that rely on different considerations than the phenotype association. We searched for a feature selection technique that uses the least number of features to avoid overfitting and achieves the highest accuracy as well. The feature selection techniques we tried are mutual information and linear SVC with L1 penalty. As illustrated in the left graph, the best score was achieved by batch 2 that consists of 441 SNPs, whose p-value of all of them raised to 10 to the power minus 3. Batch 2 achieves higher accuracy than set 0 and 1 using both linear and RBF kernel with support vector machine model. The second approach that combined both p-value technique and machine learning approach achieved better predictive capacity using both linear and RBF kernels. We mainly depend on mutual information and machine learning-based selection technique. As illustrated in the right graph, the best accuracy achieved is 93.5 using 300 SNPs. We can also note that the higher the number of features we set, the better the achieved accuracy, which supports the first hypothesis of interaction. However, the model accuracy starts to decrease at some point, which may be due to some false positive SNPs that were taken into consideration in the model. We investigated the AD correlation with the 300 SNPs that were computationally used in decision-making to find out if they are experimentally supported. Using GenoAD database, we found that 180 SNPs of them are located in 152 genes. By looking them up in literature, we found out that of the 152 genes, there are 104 unique genes related to Alzheimer disease or other related diseases, as well as 11 AD correlations with the SNPs themselves. Pathway enrichment analysis revealed that there are 146 pathways associated with these 300 SNPs, with 26 correlated pathways to neural system. We tried to use Cytoscape to do network analysis. We graphed the SNPs along with the affected genes. Nodes represented both the SNPs and genes in a network, and edge between any SNP and genes indicates the biological association. Then, for focused analysis, the nodes with more than 3 edges are filtered, which means the genes affected by more than 3 SNPs. By looking those 7 interesting genes up, we found the following 3 genes are experimentally verified to be related to Alzheimer disease. To conclude, the application of genome-wide association studies in case control setups have resulted in plethora of significant genetic variants associated with complex diseases phenotypes. Integrating machine learning-based feature selection technique outperforms the p-value statistical method. Finally, our recommendations are to increase the sample size under examination in terms of having more patients, and to apply the same pipeline to samples taken from Egyptian patients with Alzheimer's disease because the SNP print differs with different populations. We are looking for creating a personalized health report service which enables Egyptians to obtain detailed information and reports based on their genome and take appropriate action. That's it, and thank you.